Hello everyone. Today we're going to talk about polymerase chain reaction or PCR. So for any kind of genetic study, experimentation, testing in the fields of molecular biology, biotechnology, genetics, forensics, you need multiple DNA copies. So PCR, polymerase chain reaction, is a technique by which we can make multiple copies of a gene or DNA sequence. By multiple, I mean millions and billions of copies can be generated from a small amount of DNA. It is used for research and testing in biotechnology labs, and as you all might be knowing, that PCR has been quite widely used as a diagnostic tool in COVID-19 testing. In 1983, it was developed by Carrie Mullis, who was then subsequently awarded the Nobel Prize for this kind of achievement. Soon after, the Taq polymerase was obtained from a species of bacteria, Thermus aquaticus. Now, this bacteria is very special. It is found in hot springs. So, it is able to survive at such high temperatures and is able to reproduce. And this DNA polymerase is also capable of being stable at high temperatures. This machine that you're seeing is the DNA, the PCR machine, which is also called the thermal cycler. It is on this PCR machine in which the program of the reaction is fed. So, this is used to amplify DNA. This device has thermal blocks with holes where PCR tubes containing the reaction mixture can be placed. It is capable of providing the right temperatures and conditions for this kind of reaction. Now, the components of PCR is as follows. DNA template, it is the sequence or strand that we want to amplify. Then we have the Taq polymerase, which is the DNA polymerase, the heat stable DNA polymerase that we're going to use. Then primers. Primers are small DNA sequences which are complementary to the 3' strands. Now, without primers, there is no initiation. There is no initiation site where the DNA polymerase can bind and start its activity. So, they are utmost important. Then we have the DNTPs or the deoxynucleotide triphosphates. So, these are the ATGCs which are given so that the DNA polymerase is able to synthesize this new strand. Because this is an in vitro process, this is not inside a nucleus, right? Then we have buffer solution, which increases the stability and optimizes the activity of the reaction. And then we have bivalent cations like magnesium or manganese, which is used for DNA amplification. So now let us come to the procedure. There are 3 steps which are integral to all DNA amplifications through PCR. So usually PCR contains 20 to 40 repeated temperature cycles known as thermal cycles. And each of these thermal cycles has 3 of these steps. So first step is denaturation. This is the temperature at which the DNA strands are separated. So this is a high temperature, as you can see, and this high temperature leads to the melting or denaturation of DNA by breaking of the H bonds between the 2 strands. Then the next step is annealing, which happens at a temperature of 55 to 65 degrees Celsius, and this also happens for 20 to 40 seconds. So here the temperature has lowered a little bit, which allows the primers to bind to each of these now single strands. So the primers attach to the single strands. Then we have the final step, which is the extension. This happens at the temperature of about 70 to 80 degrees Celsius. Usually it's 72 degree. So here at this temperature, the enzyme Taq polymerase which we have used has maximum activity. So now it is able to synthesize new strands by adding the DNTPs from the reaction mixture. So these three constitute one cycle. With each cycle, the original template plus newly generated strands become template for the next round. So leading to exponential growth resulting in millions of copies. So here we can see the denaturation process happening where the DNA template that we have is breaking down due to the temperature and it is leading into two single strands. So breaking of edge bonds yielding two single stranded DNA molecules. Then we have the annealing process. Here the two primers attached to the two single stranded sequences themselves, but they are of much shorter length. So they are just there so that the DNA polymerase can now have an initiation point to begin. Then we have the extension. So now since the DNA polymerase can work with something because we have primers, so here at this it starts collecting the DNTPs, attaching them, and then it synthesizes new strands. And then at the end of each cycle we have the original template plus the newly generated strands, and for the next round both of these become the template strands and we have exponential growth. So this here we have a PCR graph. In this PCR graphs you can clearly see the three steps with their temperatures and the time for which each of these steps will go on. Now this is a PCR program. This is the program which is actually fed to the thermocycler in the beginning for the reaction. So here you can see the number of cycles which are fed. So the cycles also depend upon the amount of copies we want, right? Now there are a lot of applications of PCR. We have applications of PCR in the molecular biology, then we use them for research in sequencing, and then we have uses for them in genetics, then we have uses for them in forensics. There are a variety of things. So in sequencing we have used them in the past for human genome project, bioinformatics. In genetic engineering we use them for gene expression studies. In molecular biology we use them for drug discovery, DNA fingerprinting, genotyping, etc. Now at the end of this video I have a little exercise for you. You can think about these questions and pause the video here. Thank you all. I hope this video sheds some light on PCR.